Hi all, I wanted to make a video that discusses protein synthesis. And so I wanted to start off with a video for you to watch. That video is labeled under materials protein synthesis. It's essentially a 3D animation that's going to go through everything from the initial DNA code that's in the nucleus to what happens when the messenger RNA molecule is made, and then it's going to be transferred into a protein called, or into a ribosome to make proteins. All right, so watch this video first before you watch the video that I am making to put right here. And the simulation that we're going to start with is SEPUP protein synthesis. The problem that I've been having is that it's a flash player and you may not be able to view it on a remote device like a mobile device or something of that nature. So I'm making this video to go through it with you in order for you to be able to view this module. So I'll click start. The process of protein synthesis called transcription is a way in which the message that's in the DNA is transcribed, such as what a transcriptionist would do in making notes, the things that are said. It's essentially a note-taking process. So transcription's the first stage of making a protein, and during transcription, the information is coded in the cell's DNA, instructs the cell to make messenger RNA molecules out of the individual nucleotides, and it takes place inside that nucleus. And so we're going to go inside the nucleus, and we're going to look at this chain right here, and this essentially is a DNA molecule chain in animated form. And you have the amino acids, the nucleic acids that are adenine, which is A, thymine, which is T, cytosine, which is C, and guanine, which is G. Now, C bonds to G and A to T inside the DNA molecule. However, in RNA, there's something that switches up. The uracil is replaced by the thymine when the RNA is made, and it's going to occur inside the nucleus of that cell. So that RNA polymerase is making essentially those complementary strands, and then that mRNA is going to go outside of the cell. So an enzyme called RNA polymerase links the RNA nucleotides to form that strand of mRNA, which is called messenger ribonucleic acid. And when the mRNA strand is complete, the original DNA strand rezips and remains in the nucleus where it can be used over again. But that mRNA is going to travel out. So once that transcription is complete, that mRNA strand leaves the nucleus, moves into the cytoplasm, where it's going to join up with some ribosomes. All right, so that's the end of transcription. We're going to go on to translation. Okay. All right. So what translation is, it occurs inside the ribosomes of the cytoplasm of the cell. And during translation, the mRNA strand provides a template for the sequence of amino acids. Don't get those mixed up with nucleic acids, as I do all the time. And so for that sequence of amino acids in the protein being produced, with each triplet of bases in the mRNA, which are called codons, is going to match up with a complementary triplet called an anticodon. So you've got 3 of those nucleic acids, which are going to make a sequence of amino acids on a tRNA transfer ribonucleic acid. All right, the anticodons of tRNA actually match up with a specific mRNA, creating essentially a chain of amino acids in a particular sequence coded by the mRNA. It begins with a start codon, which is this right here, AUG is the start, and it ends with a stop codon, which we'll see at the very end. So what we're going to do is we're going to click and drag the anticodons onto the ribosome to match that sequence of mRNA codons. And you'd be able to do this. So remember that A goes to U, and U to A, and C to G. So where do we see it? Right over here. So I'm going to click on that and pull it right over, and it fits. And the great thing about this interactive is you see that the shapes are different so that you know that they do fit in a three-dimensional shape together. All right, so GUA would be CAU. So let's find CAU. And at the very top of that molecule, you'll see that there is an initial. And that initial is for the amino acid that it produces. So MET is methionine, VAL is valine. All right, CAU is going to be GUA. So GUA is somewhere around here, right there, histine. All right. AAA is going to be linked to UUU, which is lysine. Okay. ACU is going to be UGA. So UGA is right there. GUU is going to be linked to CAA. And so on and so forth. So a great thing about this activity is that you are going to be able to do these on your own. All right, so UGC, UACG, and that would be C to G, A to U, C to G. Histine, GGG, AC. Oh, hold on, I didn't get that one right, did I? There we go. All right, thanks y'all. You're helping me out here. All right, my brain doesn't work as fast as it used to. All right, and when you get down to the very end, you're going to have a stop codon molecule, which is UAA. All right, so that then bonds those amino acids together into a specific shape. So that chain right there is going to fold into a protein. This is a very simplified process. Really, proteins exist of many thousands of these. And that folding shape then determines, or that type of protein determines the shape of the protein. All right, and that's the end of the simulation. So, so I wanted to show you a little bit of a real life aspect of
it's a sure sign
of fireflies. I think I've got the wrong one, but we will use this right here.
how a firefly's tail makes light. First, cells in the firefly's tail make luciferase protein. Then, luciferase carries out a chemical reaction that releases light. The light comes from specialized cells in the firefly's tail. Each cell holds a copy of the firefly's entire genome. One of these genes is a set of instructions for building the protein luciferase. A molecular machine called RNA polymerase makes a messenger RNA copy of the luciferase gene. The gene can be copied many times. The mRNA attaches to a ribosome, the cell's protein-making machinery. The ribosome reads the molecular code of the mRNA to build a luciferase protein, one amino acid at a time. Multiple ribosomes can read each mRNA copy to build many luciferase proteins at a time. When the ribosomes finish reading the mRNA, the proteins are released, and they fold into their final shape. The ribosomes can read the mRNA again to make even more luciferase. Luciferase is an enzyme that catalyzes a chemical reaction between a substance called luciferin and oxygen. This reaction uses energy from ATP, and it releases light. Luciferase can catalyze this reaction again and again. The action of many luciferase enzymes acting at once makes the light bright enough to attract mates and warn off predators.